All right, here we go. Let's refute some critics on endogenous retroviruses. And we're just going to get right into it. Endogenous retroviruses. One of the evolutionary community's favorite lines of evidence for common descent. And I've done quite a few debates, quite a few discussions on this topic, debates with critics of biblical creation. I've written a roughly 200-page book on the topic, which I highly recommend. It's comprehensive. I've done at least one formal debate directly about the book, and I've discussed this question of endogenous retroviruses in great detail. So you can find the book on Amazon. You can find the links to it on the main website, standfortruthministries.com. You can also find the book linked in the description box of this video. Okay, let's get right into this because I've done several presentations on this. One presentation about the origin of RNA viruses in general was about 2 hours. I've given at least 2 lectures that were about an hour. And I've also done at least one interview that I can think of that was solely on this topic of ERVs. And that was a couple hours as well. So for this specific video, I want to focus on a line of argumentation that comes from the components of ERVs. And the components I'm thinking of are integrase, the way retroviruses can integrate into genomes of living organisms, and also reverse transcription, right? The enzyme basically that copies RNA to DNA is called reverse transcriptase. And that's actually where retroviruses got their name. Basically, they got their name from their ability to reverse the flow of genetic information. And we're going to get into that a little bit more later. The argument from essentially the structure of the ERV element, the fact that you find host genome on either side of your element with LTRs, these long terminal repeats, on flanking both ends with your gag, pole, and envelope genes, ENV genes in the middle, essentially, making up your ERV element. And that's the classic makeup of your retrovirus in general. And the evolutionists oftentimes ask, why would God create DNA elements in our genome that have such incredible sequence similarity to exogenous retroviruses, right? Exogenous means coming from outside, external viral infection, like with HIV, commonly known exogenous retrovirus. Endogenous means originating from within. And so we're born with these ERV elements or sequences or pieces of ERVs, just solo LTRs, in our genomes already. And we're going to get into how the evolutionists explain the existence of ERV sequences according to their evolutionary story. So here we go. What are endogenous retroviruses? According to the standard evolutionary story of endogenous retrovirus, an ERV is a stretch of DNA found in your DNA that got there when one of your ancestors was infected by a retrovirus. And so the greater question that I like to ask is, are these really the ancient remnants of viral infections? in the past? Or are they created units of DNA function? Both models, the evolutionary model and the separate ancestry model or special creation model, would have two very different expectations and predictions when it comes to answering that question. We'll get into that a little bit later. Notice this. Human endogenous retroviruses are expected to be the remnants of ancestral infections of primates by active retroviruses, right? So exogenous retrovirus, outside invasion of retroviruses that have therefore been transmitted in a Mendelian fashion. And so we have somatic cells, we have reproductive cells or germ cells. And therefore, if an exogenous retrovirus invades, integrates in the right place, in the right way, in a germ cell line, that can be passed on. So if that's passed on vertically, rather than just horizontally, like you would find with something like HIV, then it can be passed on to progeny. It can be passed on to the next generation. And therefore, that ERV sequence will be found in every single cell within that organism, is how the story goes. They represent about 8% of the human genome. They are a very important component of the human genome. As I just explained, ERVs are incorporated into the genome through the invasion of infectious retroviruses over millions of years, according to the evolutionary story. These infectious retroviruses have infected our distant ancestors and made their way into the germ cell. So basically, since these ERVs, every single human being has them, especially the ones that the evolutionists point to as being shared with chimpanzees in common genetic locations, genomic locations, those are fixed. They're stuck in place. What that means is every single human being has them. And according to the evolutionary story, they'd have to explain how those ERV sequences could become fixed or neutral or beneficial enough to become fixed. Fixation rates are slow. You would imagine the infection of an exogenous retrovirus that manages to get passed down through the germ cell lines, that gets passed down vertically. You'd expect that to be lost at some point. It's surely not going to be beneficial. If viral DNA inserts itself into the cell, it's more likely to disrupt genetic functionality. It's more likely to be functionally compromising to the organism rather than making the organism better, the function of the organism better. And that is especially the case if it is passed on vertically. And therefore, the evolutionists have to basically argue that over time, the infectious or the harmful part of the ERV sequence is hit with mutations, these typographical errors that occur every single generation in biological organisms and systems. And these mutations are supposed to break down through their deleterious effect, essentially, the harmful aspects of these ERVs to where they're no longer harmful and they can propagate throughout the population and eventually become fixed where everybody has them. They'll also look to co-option or beneficial mutations to explain the functions associated with ERVs that we will get into. Numerous functions associated with ERV elements and just your solo LTRs that basically just represent functional stretches of DNA that the evolutionary community want to assume are older ERV sequences that have broken down. And so now all that's left is solo components of it. What we find ERVs functioning in is determining cell types, acting as antiviral protectors. They function in viral mimicry. What's funny, even before we get into a detailed explanation of it, the way the ERV sequence is designed, we've got your LTRs, your long terminal repeats, your GAG, POL, and ENV components of the ERV sequence. They require that design. These components are required to perform their antiviral roles. They're required to perform viral mimicry. This is how they carry out a lot of their functional roles, is based on the fact that they share these sequence similarities to exogenous retroviruses. And so thank God that they were designed this way. Notice this. These infectious retroviruses have infected our distant ancestors and made their way into the germ cells. This means the next generation will have the integrated genome of the infectious virus. And this is in every one of the individual cells. And you can see all of this in their technical literature. Proponents of common ancestry believe that ERV elements are the ancient remnants of past viral infections. Some evolutionists will refer to ERV sequences as fossil viruses. Human endogenous retroviruses, HERVs, when you see the H, it's human specific, represent footprints of previous retroviral infection and have been termed fossil viruses. Notice the evolutionary base assumptions built into these technical papers in explaining ERVs. They represent fossil viruses. They're assuming they are the ancient remnants of past viral infections. Let's keep going. Evolutionists acknowledge that for the most part, these ERV sequences, the infections would have occurred millions of years ago. I've talked about the koalas today that are being infected with ERVs. These ERVs are propagating in their populations. But what's funny is your pre-existing endogenous retroviruses, they're actually working to disrupt these new endogenous retroviruses and destroying them before they can propagate enough to be harmful. And this is why, according to the model that I propose and defend, when it comes to ERV sequences, the pre-existing ones, the ones that are fixed, the ones that everybody has, we would say those are created, and therefore we would predict function. And if they're shared with the chimpanzee, or they're shared with the bonobo, the gorilla, the orangutan in this nested hierarchical fashion or pattern, well, that's exactly what we would expect if they are designed. And so these arguments that you oftentimes see from the evolutionists in terms of probability, the chances of these viruses infecting in exactly the right spots to be shared with chimpanzees if independent ancestries are true. Because remember, the evolutionists will say that humans and chimpanzees being our closest cousin, we apparently shared a common ancestor with them between 6 and 10 million years ago. That common ancestor was infected with these exogenous retroviruses, and they made their way vertically into the next generation. The next generation, the offspring had these this viral DNA in every single cell, and eventually the split occurred. Humans went one way, the chimpanzee line went the other, and so obviously they took those viral remnants with them, and they'd be in the same spots. That's how the evolutionist explains it. So they argue from probability to say that it's a very low, almost impossible chance for all of those infections to take place independently, where now we share all of these ERVs in the same spots with the chimpanzee and yet did not inherit them. But again, if these fixed functional endogenous retroviral elements, including your functional stretches of DNA, where you don't find the entire ERV, LTR, GAG, pol, ENV, LTR, host gene, then the probability argument doesn't work, because they're there for design purposes. They're created that way. Okay. But I get into it in my book, unfixed ERV sequences. They're just a small population has them. We can see with the koalas that oftentimes, actually, I would say more rare, it's a rarity, these exogenous retroviruses can be passed down vertically, but those fixed functional pre-existing ERVs, one of their functional roles, again, they act in determining cell types, gene regulation, gene expression, they act in the immune system, viral mimicry, they act in the placenta, developmental, in these developmental windows. I mean, it's function after function after function. And this specific role is basically a defense system, where if an exogenous retrovirus is passed on horizontally or vertically, then these pre-existing endogenous retroviruses, the fixed ones, the designed ones, will actually disrupt those newly endogenized retroviruses, and they'll be lost. They'll be weeded out. And so this is a good thing. Again, relics of exogenous retroviruses. So a retro element, basically an endogenous retrovirus is a type of a retro element. And a retro element is a sequence that moves around the genome through reverse transcription. And this is where I want to talk a little bit about reverse transcription. And normally we think of DNA to RNA to protein, right? DNA flows into RNA, transcription, RNA into protein, translation, transcription, translation. And so DNA flows into RNA, RNA then flows into a protein. Reverse transcription is the opposite of that. DNA to RNA to protein, RNA into DNA. And what does that in the retrovirus is reverse transcriptase. That's the enzyme that performs this job, and it basically copies RNA into DNA. And why this is important is because these retroelements, specifically the ERV, requires the reverse transcription in order to move around the genome. Okay, I want to go through this slide a little bit. A retroelement is an element that moves around using reverse transcriptase, and it doesn't necessarily have to be an endogenous retrovirus. So basically, ERVs are a specific class of retroelements, but there are different retroelements like retrotransposons, line signs, and I'm going to go over those in a little bit in another slide. ERVs are integrated proviruses. Once it actually integrates into host DNA, and with the ERV, again, you have LTR, GAG-POL, ENV. It's now a provirus. That's proviral DNA. Retrotransposons, on the other hand, have a GAG and a POL, but no ENV. And therefore, they can't spread from cell to cell because they have no envelope protein. They don't have that ENV component like the ERV does. And evolutionists assume that these have been in the genome longer than ERVs. And I want to talk about that a little bit later. So lines, long interspersed nuclear elements, or non-LTR retroelements. Remember, in the ERV, the LTR stands for long terminal repeat, and those are on either end. They flank both ends of the ERV. Then you have signs, short interspersed nuclear elements. Just to let you know, all of these have significant functions associated with them, okay? Are non-LTR retroelements and do not have their own reverse transcriptase. These sequences are transcribed and then reverse transcribed, but by another RT, reverse transcription, an ERV or line. And we've talked about this. Retroviruses can reverse the flow of genetic information, which is transcription in reverse. Retroviruses have an RNA genome that can be copied into DNA. And so if this provirus enters the germline, as we discussed earlier, it can be passed on to offspring. And now this endogenous retrovirus can spread throughout the entire population. I discussed point 4 here earlier, how mutations, according to the evolutionary story, will deactivate the virus, meaning active proviruses will cease to be produced. And then an individual may be producing active viruses, but with time and mutations, these proviruses become mutated and eventually no longer produce infectious or harmful proviruses. But those viral elements or sequences, they're still passed on to offspring. And this is where fixation, according to their model, can happen, which means a certain population will all have them, like humans today, for example. In other circumstances, they can be lost. Okay, so let's keep going here. So here's an example of your retro elements. And we have many retro elements, as you can see, such as the endogenous retroviruses. And these are tightly integrated into the genome. And these DNA elements, they actually move around the genome. They can move from one chromosomal location to another, turning genes off, turning genes on, inducing variation. That's why the Vige hypothesis that a lot of creationists hold to in advance stands for the variation-inducing genetic elements hypothesis. But your retroelements do a lot more than just function in generating variation from generation to generation, which I could talk all day about how that can explain speciation and variation since the flood with the arc archetypes that step off of the arc. And then we have these adaptive episodes, a lot of them based on environmental stimuli as well. And these front-loaded genetic elements like the retro elements, they can help with shifts in these kinds of creatures when it comes to adaptation. And you'll notice here that transposable elements, 45%. So basically between 42 to 45% of the human genome comprises retro elements. And ERVs being just one of them, the retro elements are broken up into different classes. One thing that's on my mind, though, that I want to get out before we move on is in the mouse genome, there's a specific class of retro element, and it acts in development. And so what they've done is they've snipped out that specific class of retro element. The mouse is developing, but once it's snipped out, the mouse dies. What that means is the mouse depends on that retrotransposon. There's another paper that says if we did not have these endogenous retroviruses specifically, mammals wouldn't exist. I like to exist. You guys in the audience like to exist. And so if we share endogenous retroviruses with the chimpanzee or other primates, and those retro elements function in the embryo, and without them we couldn't survive, of course we're going to share those elements with other creatures. In the same way that sedans, hatchbacks, SUVs, and vans all share engines in common. Without an engine, those vehicles are not going to go very far. Their existence would be pointless, basically. This is all evidence for common design. Why is there a hierarchical distribution of these ERV elements? Well, of course, we share more in terms of anatomy, morphology, and physiology with the chimpanzee than we do with a dog, which is another mammal, or a whale, which is a mammal. So, of course, we're going to share more of these ERV elements with the chimpanzee than we would with a dog. Now, in an impromptu debate discussion with a couple evolutionists at least, I pointed to a paper where there's actually inconsistencies in the nested hierarchical patterns that we see. This is the so-called evolutionary nested hierarchy. But the details of that might be best for another video. Okay. And so we can see these retro elements, they're broken up into different classes. You got endogenous retroviruses being one of them. The evolutionists say that these came about in the genome through ancient viral infections. And then the ERV, it has this classic design to it, the classic retrovirus design that your other retro elements don't necessarily have. So in the ERV, what we have is, so you can see here at the bottom, endogenous retrovirus, exactly what I was explaining earlier. A visual, I think, is going to be helpful. And so we have host DNA duplicated on either side of the provirus. We have two LTRs, your long terminal repeats, okay, right down here, a gag, a pole, and then an envelope, your ENV. Again, evolutionists argue this ERV is the result of an ancient infection, and the infection was through the integration, which the ERV has integrase as an enzyme. And the evolutionists look to this as evidence for external infection. We hear it over and over again in these debates. What is the integrase for? What is the reverse transcriptase for? What is the gag, pole, and ENV components of the retrovirus used for? Well, that's the thing, is the fact that these retro elements, they can move around the genome through reverse transcription. And since these DNA elements move around the genome, they are duplicated. And this has happened many times. We have many thousands of these retro elements in the genome. This would leave the same signatures that evolutionists say are there because of external infection. And so they constantly ask, why are the ERVs designed this way? What's the integrase for? What's the reverse transcriptase for? The fact that they can move around as one of their functional roles in generating variation, turning genes on, turning genes off, it leaves the same so-called scars. And again, when you hear the evolutionists say scars, genetic mistakes, junk DNA, that's evolutionary lingo that is based, entrenched in evolutionary philosophy. They assume all of this is the result of evolutionary mistakes or variation induced through different kinds of mutations, when in fact they don't know that. There's two very different starting points when it comes to explaining genetic variation in the evolution and creation model. Creationists explain the vast majority of DNA variations being the result of design diversity, created heterozygosity. And that goes for these DNA elements as well, like pseudogenes, which the evolutionists argue are the remnants of protein coding genes that were hit with mutations and broke down. Broken protein coding genes, basically. So now they're non-coding. That's an assumption. We know pseudogenes are necessary to sustain healthy life processes in the cell. I did an impromptu discussion or debate with Dr. Dan Stern-Cardinal last week on the chromosome 2 fusion, and that's what he kept saying. Well, the DDX11L2 gene, which overlaps the so-called fusion site, which is only 800 sequences, base pairs long, that's a pseudogene, he kept saying. That's evolutionary-based assumptions. There's at least twice as many of these long non-coding RNA genes in the genome than protein coding, and they are incredibly functional in so many different ways. Okay? And so it's the evolutionist that assumes that what we see in the genome oftentimes are scars, when in fact, the way these ERV sequences, the way these retro elements move throughout the genome is not entirely random. They have preferred sites. And oftentimes what we see in the genome when it comes to the mobility of these elements are akin to planes. When they're coming and they see the landing strip, they know where to land. They know how to land. And we see a lot in the genome that is indicative of where these mobile elements need to land. Because if they land in the wrong spot, that can actually result in harm, in deleterious effects. And so a lot of these elements, they are not just jumping around randomly, but they have preferred sites that they move to. And guess what? Those functional roles, internal mobility of these retro elements, requires the integrase, requires the reverse transcriptase. Since the enzyme that copies DNA into RNA is reverse transcriptase, and these retro elements oftentimes move around through copy and paste means or even cut and paste means. And with thousands and thousands and thousands of these types of elements in the genome, of course, we're going to find a lot of these signatures of mobility, internal mobility. See, the question that I frequently ask the evolutionists is, when it comes to the origin of retroviruses require a host to replicate. So what came first, the host or the retrovirus? There's a paradox, a paradox in the evolutionary model. But creationists understand that since retroviruses originated from or require a host to replicate, they must have originated within the genome. And this is what makes sense, is because within the genome, we have all the components necessary, especially when it comes to these ERV elements, the way that there's designed, that classic retrovirus design, then they would have come from within the genome. And once they spread to another organism that's not its original host, that's why you'll find that these retroviruses have very few genes, because where are they picking up these genes? They're picking up these genes from their original host or different hosts as they move around. So HIV in humans is deadly. It's a horrible disease. Nobody wants HIV. In chimpanzees, it's SIV. And wherever it goes before chimpanzees, it's harmless in its original host. But once these retroviruses go from one host to another, that's when they start burning hot and fast. That is when they become dangerous and harmful to the host. Or these pre-existing ERVs can be hit with deleterious mutations that result in disease. And that's why you'll find that a lot of times these ERV sequences are associated with autoimmune diseases and other types of even cancers as well. But because we do have examples of unfixed ERVs, then yeah, that is the result of an exogenous retroviral infection that made its way into the germline that was passed on. But now we have these pre-existing ERVs, the created ones, that are going to disrupt the endogenization process where they're not going to become fixed. They're not beneficial. And that's why the co-option argument for function doesn't make any sense, because if viral DNA randomly inserts itself into the cell of a biological organism, it's more likely to be functionally compromising than to improve the function of that organism. In the same way that if I were painting a picture and I were to blindfold myself and start applying random brushstrokes to the painting, I'm not going to make it any better. I am actually going to make it worse. And so we've got these various classes of retroelements. We've got these retrotransposons. Okay. And these have, as you can see, two LTRs, gag, pull, but they have no, as I went over earlier, envelope, which means they can't make a particle since they lack this gene, this ENV gene. And your virology community, they recognize this paradox about where retroviruses came from. And since they believe ERVs are the ancient remnants of retroviruses, they have to explain the origin of retroviruses. And many prominent virologists have actually argued that these retrotransposons are the progenitors of retroviruses. And so retroviruses originated from within the genome. The escapee hypothesis, there's variations of it. Your evolutionary virologists are recognizing this. And so they'll argue that they were probably around before retroviruses. And then at some point, they would have captured an envelope gene from the cell, because these components, these elements are in the cell. All it takes is an error or disruption in the packaging process, and boom, you get a virus. And then it can move from host to host. It can escape the cell. And so these virologists will say that they captured an envelope gene from the cell and became what? Became a virus, which means it can now go from cell to cell, since it now has an ENV. And this is why the Cratius has the best explanation. And again, it's an answer to, well, why do these ERV sequences resemble retroviruses, exogenous retroviruses? Well, if these retroviruses came from within the genome and these ERV sequences and these retrotransposons pre-existed these retroviruses, these harmful retroviruses, because they were designed and created in those positions, and then they were designed to move around, there's probably a lot more mobility in the pre-flood world. And also for your first 1,000 years after the flood, where these massive shifts in environment are occurring. And so more adaptation is required. But also over time, we've accumulated a lot of mutations. We accumulate 100 new mutations per person per generation, most of those deleterious. And when they hit these mobile elements, they may disrupt their functionality. We see degeneration. Things aren't getting better. ERV elements aren't getting any better. Okay. So yes, with so many retroelements in the genome, are we surprised that we see these signatures that the evolutionists say are there because of outside or external infection? No, you get the same signatures from internal mobility, from the function that is designed in these elements that are integrated, tightly integrated into the genomes of biological organisms. And eventually, as some evolutionists even would agree with, but obviously they wouldn't agree with my ultimate conclusion, but the conclusion that best is supported by the data is that these retroviruses came from within the genome rather than outside, because retroviruses require a host to replicate in the first place. So again, here's your ERV sequence. You got host DNA on either end, duplicated on each end. LTRs, gag, pull, and ENV, which are retrotransposons. actually lack. So here's questions that I've answered in the past. In a nutshell, why are there ERV sequences shared between the genomes of organisms? Well, of course, given their functional roles, if they're created that way, and that is a prediction, a testable prediction. The gold standard of science is making testable and falsifiable predictions. And in my ERV book, I predict that your fixed ERV sequences that are found in every single human being, with many of those shared with the chimpanzee, and in the chimpanzee they're fixed as well, they are created and therefore they are functional, beneficial to the organism. Mostly not disease causing, but again, deleterious mutation accumulation over time can take a functioning or a beneficial endogenous retroviral-like element to something harmful, to something that is now bad, simply due to the degeneration of the genome and all of that comprises the genome, including your pseudogenes, including your ERV sequences, your ALU sequences, your retrotransposons, lines, signs, so on and so forth. If viral-like sequences in the genomes of organisms are functional, why do they bear similarity to viral genetic material? This goes for the reverse transcriptase, the integrase, the GAG, the POL, the ENV, the LTRs. They're all there for a reason. They're required for viral mimicry. Here's the thing. When the cell is under stress, and I've talked about this, I talk about this in my book, I've discussed it in my debates, the cell, when it's under stress, what happens is transcribed ERV elements, they actually give the cell the appearance of being invaded by a viral infection, which as a result targets that cell more destruction by the immune system. And some tumor cells can evade detection of the immune system, but because of this mechanism, this incredibly designed mechanism, which is actually mediated by a specific protein that many have termed the superhero of the genome, this superhero protein, P53, is a way for those tumor cells to actually become detectable by the immune system. And as a result, this allows the immune system to basically clear those tumor cells from the body. This is an amazing ability. And many scientists right now, you can read lots of papers on this. Many researchers, they're working on specific drugs that looks at this P53 super protein. And these drugs are being developed to basically stimulate P53 activity and the transcription of these ERVs, these pre-existing ERVs that are there for various reasons, this being one of them. And this is all for anti-cancer therapy. This is all evidence of forward thinking. And so we have ERV elements basically playing a role in tumor suppression through viral mimicry, which means these ERVs require, in order to carry out that role, it wouldn't make any sense to perform a type of mimicry function if they didn't look like what they were trying to mimic. It would be like these undercover agents in the FBI infiltrating some crime family or the mob. Well, they got to now look the part, because if they show up dressed in their cop uniform, well, that's not going to be very smart. And so this is just one of the functional roles. It's the same thing with these thousands and thousands of ERV elements that are there to fight off external invasions. And they're able to do this because of the way that they're designed. And I've said it over and over again. It's redundant, I know, but these evolutionists won't stop asking this question. Why do these ERV elements, Dr. Dan Stern-Cardinal, who dodges our answers, my answers specifically because I wrote a specific book on it, the properties, the components, the structure of the ERV elements are required for their roles in antiviral protection, in viral mimicry, in moving around the genome. They require the reverse transcriptase. They require the integrase, especially if through cut and paste or copy and paste, moving from one chromosomal location to another, everything about it, the ERV element, is required for that which they were designed for. You're going to get the same signatures that you would get if it was external infection. But the key difference is we understand that we have these ERV elements. They are in abundance within the human genome specifically, and they have these many, many functions, including mobility that generates variation from generation to generation. And this is amazing stuff. I oftentimes say what the evolutionists thought were their best lines of evidence actually turns out to be some of the best evidence for separate ancestry. And that also goes for these retro elements. What is the best explanation for the nested hierarchical distribution of endogenous retroviral-like sequences in primate genomes? Again, if these are created units of DNA function, which the evidence suggests, highly suggests, and of course we're going to share more with the chimpanzee than we would with a dog or a whale. We already share more in terms of anatomy, morphology, physiology, and overall genetics anyways with a chimpanzee than a dog. Just stand afar. We clearly share more with the chimpanzee than we do with an old world monkey, a lemur, than we do with an orangutan. So this isn't a surprise. There is a natural nested hierarchy even in modes of transportation. This wasn't done intentionally. It's not like car engineers said, you know, we're going to build all these different kinds of vehicles, and we are purposely going to try and mimic the biological world by designing and nested hierarchical patterns. No, this was done accidentally. The fact that sedans share more with each other, of course, you can group all these different kinds of sedans in their own group called sedans, but sedans share more with SUVs than they would to a tractor trailer. And then sedans, SUVs, and tractor trailers share more with each other than they do with a plane. And so we have these groups within groups patterns that manifest when we start looking at these different features within modes of transportation. And it's the same thing when it comes to the biological world. This didn't have to be the case, and yet it is. And so if what I am saying is true, that these reflect created units of DNA function, then of course there is going to be a natural nested hierarchical distribution of these ERV elements, but we would also predict inconsistencies. And we've actually found many inconsistencies, which I oftentimes point to in debates. Can the separate ancestry model explain the data better than the common descent model? Yes, especially because we can make testable and falsifiable predictions, which is the gold standard of science. I have laid it all out in my book, in my lectures, in my discussions and debates. These fixed ERV sequences, they are going to reflect design through function. And that's why we see in your isolated cases of unfixed ERVs, the pre-existing ones are actually working, hence functional, to disrupt the endogenization that has taken place. We see this in the koalas. It really is amazing. So in the early stages of development, we know that humans develop what's called a placenta. And these ERV elements function in the placenta. It's necessary for exchange between mother and baby. Without this placenta, humans, we wouldn't exist. And during pregnancy, when this develops, it's a temporary organ. And again, this organ, this structure, it is necessary for a healthy pregnancy. It provides exchange between baby and mother, as I've said. This includes nutrition. It also helps get rid of waste. And for the placenta to work, obviously, this important structure needs to be connected to the baby. And guess what? An endogenous retrovirus is actually key to embryological development and placental function. The placenta secretes a protein that binds it to the embryo, which actually keeps the two connected for the next several months of development. And that reminds me, there's actually very specific ERV sequences that are only active in certain developmental windows. But once those developmental windows are over, the ERV is turned off again. It's rendered inactive and can even be repurposed during the life of the organism, where it can be called upon again, maybe for an adaptive episode of some kind. And so these evolutionists that strictly look for transcription or activity, they are misunderstanding the basics. They are demonstrating their ignorance in the fact that many non-coding sections of our genome, many non-coding RNA genes like pseudogenes, for example, ERVs included, endogenous retroviral units, are only active under certain circumstances. And this includes development. These ERV elements are actually safeguarding the baby from viral invasion. Again, I would predict that the way they're designed, the elements, their components, are necessary for that role. A lot of these ERV elements are actually acting as antiviral programs, just like our computers are built with Norton antivirus, antiviral programs that no computer should be without. And we do not want to be without viral-like elements, including ERVs. That is a fact. And the evolutionists can't explain it. Co-option doesn't work. It's a fairy tale. No empirical evidence for that. Again, if viral DNA just randomly inserts itself into the genome, it is more likely to disrupt genetic functionality. That's what we see. HIV. When the HIV exogenous retrovirus infects, nothing good comes about that. So again, the placenta, going back to the placenta, it actually secretes a protein that binds it to the embryo, which keeps the two connected for the next several months of development. As I pointed out, this has to be the case for the placenta to work. And the DNA that actually makes up that protein, it's very similar to a region of a retrovirus that allows the virus to attach to its host cell. And so these pre-existing, these internal functional endogenous retroviruses, again, have these sequence similarities to exogenous retroviruses in order for them to systematically carry out many of their roles. And if your retroviruses came from within the genome, came from healthy ERV elements, then guess what? Of course, they're going to share these similarities. The evolutionists have this backwards thinking. They keep asking, why do the ERV elements resemble the retroviruses, when they should be asking it the other way around, since retroviruses actually originated from within the genomes of living organisms. And this has to be the case because retroviruses require a host to replicate. I've said this 5 or 6 times, but the evolutionist doesn't seem to get it. They also don't have a response to that question. They do not have a response to that paradox. And so without endogenous retroviruses, the placenta, which is absolutely critical to pregnancy and embryological development, would not work. We wouldn't exist. I like to exist. Hopefully the viewer likes to exist. We wouldn't exist without these endogenous retroviruses. These functional DNA elements, they are necessary for life. Of course, we're going to share. I wouldn't be surprised if nearly all forms of life shared these endogenous retroviral elements, including your functional stretches of DNA, like your solo LTRs, that, believe it or not, are also functional and necessary to sustain normal processes in the cell. Okay. So again, they're essential to life. And not only that, these ERVs, they don't just assist in embryological development and embryonic immune systems. They also work in our immune systems after we're born. So they're working in the immune system during these developmental windows, and they're also working in our immune system during our lives. And our immune system pathways are actually dependent upon enzymes generated by what? Endogenous retroviruses and ERV-like elements. The evolutionists are without an answer. That's why they don't have a sophisticated model, and that's why they continue to ask these same arguments. Why does the ERV have LTRs, GAG, POL, ENV? What's the integrase for? It's all necessary for their functional roles. The integrase is necessary for their mobility throughout the genome. It's the same signatures that we'd see, and they're the ones that assume that these are so-called scars oftentimes. when in fact the movement throughout the genome by these retro elements are not random. They're non-random. They have preferred sites that they move to. But over time, since it's been thousands of years, since the flood and since creation, there has been deleterious mutation accumulation. There's been degeneration of these mobile elements. But there are thousands of them. And so a lot of these signatures are expected to be there simply because of their functions externally or internally, not externally, like an HIV virus coming in or exogenous, a retroviral infection in the past. No, the evidence is not on the side of the evolution model. So another thing, these so-called ancient remnants of past viral infections, remember, that's the evolutionary based assumption. That's evolutionary philosophy. They are safeguarding our bodies against microbes and other viruses. These viruses come in externally, and these pre-existing functional and fixed ERV sequences or other classes of retro elements, they're there to fight off these viruses that are invading and looking to cause harm and disease. There is a lot of significance to these ERV elements. It really is amazing. And so you can read all about their function in the placenta. Here's an interesting read for sure. I've gone through all of this before. And again, here the detail, the pol gene encodes viral enzymes such as reverse transcriptase and integrase that are needed for transcription of the viral RNA into double-stranded DNA, and the integration of the DNA produced by reverse transcriptase into the host genome, respectively. The way they're designed, these ERV elements or these other classes of retro elements, the way that they can move around the genome internally is based on their design. It is required. And it's not just, of course, I've done whole shows on this, whole debates on this. It's the entire junk DNA paradigm that's been overturned. But there is a couple other slides I want to get to as I wrap this up. And so junk DNA overturned, paper after paper. Target site duplications, as these evolutionists point to with these ERVs. However, several TEs, transposable elements, still are active in other organisms. We can say transposons and transposition are one of the evolutionary forces, of course, I got to throw that in there, in which the target site duplication plays an important role. Notice this, we can say the process of target site duplication induces what? New variation in nature. Exactly. And the further you go back in time and the less deleterious mutation accumulation that has taken place, you can have variation induced quickly because of these front-loaded DNA elements. It's an amazing study, and I'm really enjoying digging deep. Notice this, 2022, switching sides, how endogenous retroviruses protect us from viral infections. So these evolutionists want us to believe that millions of years ago, thousands to millions of these exogenous retroviruses invaded, integrated, got passed on vertically through the germ cell lines, and eventually evolved the ability to fight off that which they were. I mean, that is some storytelling. That is some evolutionary philosophy right there. That's like a robber breaking into my house. I wake up at night and the robber's like, you know, I like it here. I'm just going to move in. I'm going to make myself at home. I'm okay with it. I mean, this is nothing but imagination. You know, it makes sense that God would have front-loaded the genomes of living organisms with many kinds of functional elements that would fight off viruses, that would fight off harmful agents that are trying to invade. Notice they even admit it, long disregarded as junk DNA. No longer junk DNA, guys. Or genomic dark matter, endogenous retroviruses have turned out to represent important components of the antiviral immune response. And when they bring up co-option, or beneficial mutation accumulation. I say cool story, bro, because this is evolutionary philosophy. They don't have an example of a harmful retrovirus being passed on vertically, being hit with a series of mutations, and then becoming functional, especially functional in something like the embryo, where without it, we wouldn't exist. Now, cool story, bro. Again, if we as humans share more of these ERV elements with chimpanzees than we do with a mouse, it's not a big surprise. Just stand back and look at the anatomy and morphology of the chimpanzee and the mouse. And it doesn't take a genius to realize that in terms of genotype, we are going to share more with the chimpanzee than the mouse. But again, there's still inconsistencies in the so-called evolutionary nested hierarchy. And that's why in the past I've talked about convergent evolution. I've talked about the Y chromosome dissimilarity between humans and chimpanzees that actually make humans and gorillas closer to each other in the Y chromosome than humans are to chimpanzees. Notice just in your primates, apes and humans, because the evolutionists, they classify kingdom, phylum, class, order, family, genus, species, right? Linnaeus classification. They classify humans as human apes, and then your gorillas, orangutans, chimpanzees, bonobos as non-human apes. Well, it turns out that we share more ERV sequences with the great apes than we do with your lemurs or your New World monkeys. It's the same case. We would predict a general nested hierarchy because just stand back. We share more in terms of morphology and anatomy and overall phenotype with a gorilla than we do with a lemur. This really shouldn't be that difficult. Your sedans share more with your SUVs than your sedans do with your airplanes or your bicycles. Inconsistencies with the hierarchy. Notice this: a HERV-K provirus in chimpanzees, bonobos, and gorillas, but not in humans. These observations provide strong evidence that for some fraction of the genome, chimpanzees, bonobos, and gorillas are more closely related to each other than they are to humans. And there's many, many examples of this. That's why there's something called incomplete lineage sorting. I mean, depending on what you're looking at in terms of genes and DNA elements, it might turn out that humans are closer to gorillas on that specific tree than they are to chimpanzees. Notice this, what I've been saying. Just basics. This is the basics of transposons. The ability of transposons to increase genetic diversity together with the ability of the genome to inhibit most TE activity results in a balance that makes transposable elements an important part of evolution, of course. But we understand what that means is just adaptation that creationists believe in. And gene regulation. Notice this: in all organisms that carry these sequences, they're functional. A lot of them are generating diversity. They're moving throughout the genome. That mobility requires the integrase. It requires the specific components that make up the ERV sequence, these retro elements. And these evolutionists are asking questions. And these questions demonstrate that they don't understand the basics of retro elements to begin with. HERVs appear to play important roles in physiology, fetal development, and human evolution. Human endogenous retroviruses have recently been suggested as mediators of normal biological processes, such as cell differentiation. Guess what? The chimpanzee has cell differentiation. The human has cell differentiation. And so, of course, we are going to share similarities in terms of DNA elements that are involved in cell differentiation between these creatures. Regulation of gene expression, gene regulation, determining cell types. These are all aspects of your chimpanzee species and your humans. So, of course, we're going to share these elements. It's common design, and it should be basic. I could spend all day going over these many papers. Taken together, our findings suggest that HERVs behave like normal cellular genes and are a permanent component of the transcriptome of a cell. Retroviral promoters in the human genome. These data suggest that ERVs may regulate human transcription on a large scale. This isn't something that's small scale. This isn't spurious. Endogenous non-retroviral RNA virus elements evidence a novel type of antiviral immunity. It's all over the place, guys. Screenshot this, look up the sources yourselves. And any time that they invoke co-option, they're not actually providing evidence for it. They're basically just saying, well, you know, these functions were co-opted, but no evidence for it. It's the evolution story integrated into these technical papers. Scientists identify new beneficial function of endogenous retroviruses in immune response. Guys, we are in the beginning stages of understanding the DNA language. And as creationists, we can predict as time goes on and more sophisticated ways of determining function within the genome come about, we're going to find more and more function for pseudogenes, ERV elements, ALU sequences, all types of retro elements, lines, signs, and LTR retro elements, non-LTR retro elements. But the evolutionists, they are reluctant to make these kinds of predictions because they require genomes of junk for common descent to be more likely. It's not likely. Paper after paper. Transposable elements have had a substantial and sometimes beneficial impact on host genomes in several, not just one way, several ways. Transposable elements are a prolific source of tightly regulated, biochemically active, non-coding elements, such as what? Transcription factor binding sites and non-coding RNAs. As we have summarized, I talked about this one earlier, but I want you to notice this part at the end of it. If the accidental infection, see, that's evolutionary philosophy. They're assuming that there was an accidental infection millions of years ago in order for mammals to develop the way that they have and evolve the way that they have. But that's storytelling. What we know is these ERV sequences exist. Without them, we wouldn't exist. And so the chimpanzees exist, humans exist, mammals exist because of these ERV sequences. Of course, we're going to share them in the same way that modes of transportation share an engine and share many different kinds of features, thousands of features. If the accidental infection of a mammalian ancestor by an exogenous retrovirus had never occurred, the placenta and the mammals that produce it, including humans, would never have existed. Do we need any better evidence? Yeah, a lot of the evolutionists won't tap out, but they're cornered. Guys, they're playing defense on endogenous retroviruses. They're playing defense on the chromosome 2 fusion. They're playing defense on pseudogenes. They're playing defense on all of the arguments that they used to think was their strongest lines of evidence for common descent. Our results suggest that a subset of transposable elements are important for gene regulation in early mouse development. Oh, creationists, tell me why we share as humans a certain portion of our ERV elements with mice. Well, if mice require TEs for gene regulation in development, and so do humans, then of course we are going to share those. Again, that's evidence for common design. The differentiating way to determine what is true, the design hypothesis or the common descent model, is function. We predict function. These evolutionists predict junk. We expect genomes of treasure. They expect genomes of junk. And yet what we're finding is more and more activity in the genome, and there's significant overlap with what ENCODE found with the activity that is somewhere between 60 and 80%. There is significant overlap between that activity and ERV elements and pseudogenes, suggesting that they are active for a reason because they are functional and they are serving purposes. And we know of many purposes. 2022, we're having papers come out 2023, we're having papers come out showing more and more function. Overall, our results provide direct evidence for retrotransposons in actively shaping cell type. So, believe it or not, your chimpanzees have what's called cells. Your mice are made up of cells. We're made up of cells, 100 trillion cells. And so, if we're also made up of retrotransposons that are actively shaping cell type and species-specific chromatin architecture, then guess what? We are going to share those with chimpanzees, gorillas, orangutans, lemurs, dogs, whales, mice, because they also have and require DNA elements to shape cell type. It shouldn't be that difficult, guys. Transposable elements contribute to transcription regulation. Guys, pick up my ERV handbook. There is dozens of papers right from the scientific literature confirming separate ancestry. Now, when you read a lot of these papers, yes, they'll just throw co-option in there. They'll just throw engines of evolution in there. But they don't provide any evidence. It's evolutionary-based philosophy. When you go in there, chew the meat, spit out the bones. Just spit out, toss aside the evolutionary philosophy, look at the data. Oftentimes, these papers have conclusions that are riddled with evolutionary-based assumptions. The reason why these elements contribute to transcription regulation, they contribute to determining cell types, they act in the embryo, they act as antiviral protectors, they're involved in gene regulation and gene expression, is because they are designed that way. And therefore, the reason why we share so many with the rest of the biological world is because of common design and not common descent. Remember what I said earlier about cancer therapy? Notice this. 2021, endogenous retroviruses are emerging as promising therapeutic targets in cancer. Notice emerging and notice promising. That means there's more research to be done. We are in the infancy of understanding the DNA language. It's like if I were to be hired to go over to Russia or Germany to act as a proofreader. I can't speak the languages. I'm not going to be a good proofreader. I am not going to be able to determine that which is a mistake and that which is not a mistake. And this is similar to what scientists have been guilty of and are still guilty of, is when they study the genomes of living organisms, they assume that certain genetic sequences, genetic units, and genetic elements are mistakes, are evolutionary leftovers, are fossil viruses. But in fact, they are not. They just understand so little of the DNA language, in the same way that I understand so little of the Russian language, that I would not make for a good proofreader. Now, we are understanding more and more about the genome, and the more and more we understand about the genome, guess what? We are understanding that which scientists used to say are genetic mistakes are actually not genetic mistakes, because they are necessary for the cell. They are essential for a life. They are life genes, genes and elements that are there. And if they weren't, life wouldn't exist. Promising means there's a lot of research to be done. Far from being junk DNA, the pervasive retrotransposons that populate the genome have a powerful capacity to influence genes and chromatin. This is fascinating stuff, guys. HERV-H, a specific human endogenous retrovirus, can modify the higher order 3D structure of chromatin during early primate development. And again, I can go on and on and on. ERVs have become integral parts of immune defense mechanisms and help to fight off invading viral pathogens. What about the solo LTRs? Guys, they represent functional stretches of DNA. Here's just a couple papers. Endogenous retroviral LTRs as promoters. So we have all these genetic switches in the genome that are there to initiate and promote protein synthesis, the protein synthesis process. DNA transcribed into RNA translated into protein. Function after function. Notice this: endogenous retrovirus long terminal repeats as ready to use mobile promoters. Amazing stuff, guys. Amazing stuff. And so we are going to wrap it up there, though. And to summarize, guys, the way the ERV elements are built: LTR, GAG, POL, ENV. Some other retro elements like retrotransposons, they lack the ENV. And even many prominent virologists have argued that retrotransposons are the progenitors of retroviruses, and that retroviruses probably came from the cell, which is what we're saying. And so the evidence is on our side. The way that they're designed, the way that they're built, their specific enzymes and components like reverse transcriptase and integrase, they're all there for a reason. They're necessary. It's not evidence of external infection. It's evidence of the functions that they are designed with. And this includes their mobility throughout the genome, where they can move from one chromosomal location to another. The integrase is required for this. Everything about the ERV element is required for them to systematically carry out their numerous functional roles. And so with that, guys, thank you for sitting through this presentation, just over an hour. It's a lot of information, I understand. It's a technical subject. And so please, I highly recommend picking up the Endogenous Retrovirus Handbook. Check out my debates, where you can watch firsthand these evolutionists, these critics, utilize these arguments that I'm debunking in live debate, where I debunk them in live debate. And so at this point, guys, when it comes to ERVs, the argument from endogenous retroviruses, they're just repeating the same arguments that have already been debunked by myself over and over and over again. With that, thank you so much for watching this video. Please share around this content. The truth is so incredibly important. God bless.